Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. I'm very excited to bring you brand new episodes. To start, I had the pleasure of hosting Dr. Richard Premont. First, we discussed his career trajectory and then moved on to record a series of episodes in which he shared the history of the significant discoveries that shaped our field. I had such a great time chatting with Dr. Premont. I hope you'll have as much fun listening. Before we dive into this episode, I would like to take a moment to thank our Dr. GPCR Ecosystem partners, Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Please mark your calendar for our next symposium held on Friday, November 17th, on the topic of GPCRs in immuno-oncology. The symposium is free, but you must be a Dr. GPCR Ecosystem member, which is also free. For our last event of the year, we will be hosting a roundtable and discuss GPCRs and their role in immunology and oncology, and current therapeutic modalities in this context. The GPCR retreat is coming up November 2nd to the 4th. Although registration is closed, the organizers are looking for the event's next logo. Please visit gpcrretreat.org and get more details on the logo contest. Wouldn't it be fun to have your design associated with this iconic meeting? And now, let's dive into this episode. Welcome back to the Dr. GPCR podcast. Last week, you heard the first part of our conversation with Dr. Richard Premont. This is the second one. I hope you're going to enjoy it. Let's dive in again.
One kind of funny story I want to tell. So I was just about to finish my graduate work, and a paper came out from across the street. So I was at Mount Sinai. This is from Columbia, and it was Linda Buck and Richard Axel reporting the olfactory receptor family, because they didn't say here's the one olfactory receptor. It's like those 19 receptors in their paper, plus genomic blots showing that there's like a zillion more. Now we know it's like 800. Yeah. 400, something kind of. I mean, rodents have more. It's like 800 in mice. So the interesting thing about this paper, right? So now here's a new sensory system that people didn't necessarily know was G protein regulated. So now, poof, that's obvious because now that's what they are. But it, really changed the number of receptors because this huge family. The interesting thing is if you download that paper, go to the very end and look at the submission date, right? So this is 1991, back in the days of paper journals. There is no online anything, right? 3 weeks between submission and not acceptance, coming out in paper in the bound journal. 3 weeks.
Wow. Okay.
So that's what happens when you have. This was back when Cell as a journal was owned by and run by a guy named Ben Lewin. And when he made a decision, he made a decision.
Interesting.
And so that's when we had an editor who could edit. And it's like, yeah, I know this is exciting. This is interesting. Yes, we got to get this out now. So
3 weeks. Good. That's a record, I think.
I
don't know. Less than 3 weeks.
Yeah.
Less than 3 weeks.
There's a revision thrown in there too. It took 4 days for the revision.
There's a potentially a reviewers involved, or revision, and think of, there was no internet at the time, or at least very limited.
They're sending paper copies in the mail, and somebody's got to retypeset it, and
exactly
the redactor is going to go. Yeah, 3 weeks. Go to the printer. So, yeah. One other kind of funny story while we're on the kind of purification cloning phase. So there are a lot of groups that are trying to do this, right? So I'm not going to, I apologize to the people I'm skipping over. But one kind of funny story I wanted to mention is there was one group in particular that was looking at trying to purify the beta-2 receptor in particular, and that was a guy named Craig Venter at NIH, and he'd worked with tethered ligands and things, and he had antibodies to receptors. He did some good progress, but then Bob's paper came out, and it kind of took the wind out of his sails. So they kind of changed the lab a little bit and started to look at, cloning other receptors. And in one of those kind of really weird twists of fate, they're very, very early adopters of automated sequencing machines. So they had one of the very first ABI sequencers because they were at NIH. I guess they had the budget for it. So if you have a hammer, everything's a nail. So they had the sequencing machine, and they wanted to keep it busy because they invest a lot of money.
I mean, if you buy it, you might as well.
and receptors to sequence are hard to come by. So they started to do this thing that a lot of people kind of ridiculed them for at first, but it was I think really kind of brilliant. They just took a cDNA library and played it out. So they just pick one phage at a time, one clone at a time, and then take that clone, grow it up, and sequence the ends to get and what they called it was an expressed sequence tag. So they did this for thousands and thousands of clones, building up a library of information about genes that are being expressed, right? So not knowing what they are, just knowing that there's a thing and it's expressed. And then when the Human Genome Project started as a government project, Craig left NIH, founded Human Genome Sciences and the Institute for Genomic Research, and kept doing the same thing with ESTs and also sequencing fully through things and patenting everything. They patented every gene on the planet. But they also, technically, I think they beat the Human Genome Project for sequencing the human genome. So basically, we have, in a way, Bob to thank for that. Because he kind of, Craig kind of left the field because it was getting, it was getting away from him, and he just moved into a different direction and was enormously successful. And it's Bob's fault.
Love it.
Yeah. So as I said, the receptor cloning kind of had a slow start because the people were cloning things by homology to the couple that had been found, and they're starting to find subtypes.
Exactly.
And so that was basically the adrenergics and the muscarinics, and then this one oddball that turned out to be a serotonin, right? But where's the ACTHs? Where's the TRHs? Where's the chemokines? Well, that was solved in 1989 when a group in Brussels, headed by a guy, Gilbert Vassart, took all the sequences for all the GPCRs that were known at the time and lined them up. It's like we say, well, rhodopsin looks like the beta receptor, and there are residues that are similar. But he looked at it very carefully and identified all the regions of the receptors that looked the same, right? So your famous, there's like GNXXV and membrane span 1, and the salad box and 2, and the dry motif and 3, and all the way through to NBXXY, right? And they took those sequences and made degenerate oligonucleotides. So up until that point, people had making degenerate oligonucleotides to actually radiolabel them and do hybridization to screen a library. They didn't do that. PCR, polymerase chain reaction, had just come out, and PCR as a concept is really transformed the world, right? So kind of a digression on PCR. So PCR started with using a regular DNA polymerase, doing a reaction, and then dunking your tube in boiling water, taking it out, putting in fresh enzyme because you just killed it, step after cycle after cycle after cycle after cycle. So there are no machines; it was all done by hand. And among the first people to get it to work was a guy named Tom Caskey at Baylor, who actually figured they had like a machine that could do this.
Wow.
Thing. It was before Taq polymerase, right? So thermostable polymerase. So anyway, but by this point, thermostable polymerases had come out. Machines had started to arrive. So it was a technique that you didn't have to be crazy to do. If you can imagine the hands-on work to get that to work at the beginning. But it works. Okay. So what Vassart did was he applied PCR to GPCR discovery. So he took these primers that were able to bind to maybe lots of different GPCRs and just amplified. And then they used dog thyroid cDNA as their source, and just amplified, took what they got, subcloned all the bands, sequenced them, And in their first paper, they had 7 GPCRs that they found, right? Now, at the time, right, the primers are based, the primers are biased toward the receptors you already know, of course.
Yeah.
So you get a lot of things you already know. So 3 of the 7 receptors they found eventually turned out to be known. They were immediately known. And four of them they sequenced and complete, did northern blots and all that kind of thing. But they didn't know what they were. They were published as orphans. So this is the first intentional orphan paper where somebody went out of their way to identify orphans as GPCRs to see what's out there. They were specifically looking for the TSH receptor, which they did not get. Mm-hmm. But what they did get was adenosine A1 and adenosine A2 receptors, and a serotonin receptor that was not one of the ones that was known, plus one of your favorites, CXCR7.
Yeah. You said dog, and I was like, okay, I think I know where this.
I remember that one. Yeah. So that really opened the floodgates, because now you had a technique that didn't rely on you necessarily knowing anything about your receptor other than it's in the family.
Yes.
Right? So you could take different combinations of primers that are biased in different ways, and a lot of people did this and found all kinds of receptors. So I think one of the ones I want to mention was that there was a group in Canada, Brian O'Dowd, who was a Lefkowitz alumni, with Hiram Nizhnik, and they did the same kind of thing after this. One of the things they found was D2 long, right? But they also found a lot of other receptors, and in one of their papers that came out in 1994, Adriano Marchesi, who you've talked to, was the first author on that paper. That's the paper where they first say an orphan is GPR number.
Yes.
Right? So that's where that comes from. But the O'Dowd group, they identified dozens and dozens of receptors, and then okay, so now you got these receptors. Now you have a different problem. Instead of having a ligand and a function, a hormone and a physiological function, now you've got a sequence.
Yeah.
It's the days of molecular biology. You can express it, but now you need to start searching what's the ligand.
What does it bind? Exactly.
And then for function, all the other G proteins are starting, they're also being cloned. So now we knew that there was, GQ, G11, G14. You knew that there was G12, G13. GQ is very quickly assigned to phospholipase C regulation. Mm-hmm. So that was good. It took quite a while for 12 and 13 to get assigned to activating RhoA, and that's also, a lot of that was done by the Dallas group. That's a lot of that's Paul Sternweiss. So at some point you kind of had all the G protein effectors, right? But there's a lot of potential ligands that you have to screen. So people would look, well, where's this orphan expressed? And it's like, oh, it's high in, kidney, but it's low in liver and what have you. I'll try to guess what it might be. And as more and more receptors started to be identified with what their ligands were, you could start to predict, well, wow, this looks a lot like all the other peptide receptors. It's probably going to be a peptide. The things like the TSH, LH, FSH receptors are like the adhesion receptors, have these huge extracellular domains. So as soon as you see one of those huge extracellular domains, it's a glycoprotein hormone probably. Yeah. And then all the small molecules, the catecholamines, the epinephrine, dopamine, and the serotonin, all those receptors kind of clustered together too. And the oddballs are things like the one orphan that I published and cloned turned out to be an LPA receptor, which is basically a lipid. The lipid receptors, it took a long time for them to be twigged because binding assays are notoriously difficult, and you weren't sure what they're coupled to, and it was and it's like things are activated by components in serum, and it's just the background is high as kind of a mess. But in any case, it's over time. You could start to take a guess as what it is. Even today, the orphans, you have an idea that this is going to be a peptide of some sort, but you just don't, no one's found what it is yet. But it was an amazing age where just like now, where structures coming out all the time, is like sequences are coming out all the time. Sometimes identified with their function, sometimes not, and it was kind of interesting days. So we had when I was in the Lefkowitz lab, it was I don't know if it was GPCR-DB when it had just started, but some site had started to collect all the sequences, mm-hmm. and they had an alignment of all the GPCRs, and we printed it out on like 100 pages of paper and then taped the pages of paper together on so you could go
Wow.
and you see, and it turned out that, if you look at it, you could see that for all the GPCRs, again, which, and this was all family A, right? The only residue that was 100% conserved is the R in the dry motif. And there was like one
Wow.
that didn't have it, and I was like, I'm sure that's a sequencing mistake, because but that's the one residue is that arginine. Everything else varies, but within families, there's clusters of conservation.
Yeah.
Yeah.
How interesting. So we went from using tissues to observe function to then
Yep.
purify the components to then put them together to then sequence them and clone them. And then when we got to cloning and we started identifying more and more receptors, then the other end became a problem, is that we knew what the receptor was, but we didn't know necessarily what the ligand was.
Right.
So I think we just went full circle here.
But early on, right? So we had a list of all the endocrine hormones, all the autocoids, all the lipid factors, all the neuromodulators. So we had a list of things to go through, but for the most part, we've exhausted that list, right?
Yes.
So now the problem is you've got a ligand that you don't know what it is, and I think my bias is having I spent a lot of time thinking about metabolism, and people are finding a lot of receptors that are responsive to metabolites. I think a lot of the orphans are probably going to be responding to metabolic products, but time will tell. Yeah.
Love it. Love
Um,
it.
okay. So far we've basically been ignoring the visual system.
Yes.
And you'll notice this is not an uncommon thing. I think that the visual signaling community, I think, got very slighted.
Yeah.
A lot of a lot of the things we know about the way these signaling systems work, they found first, and being second and noticing the similarities counted for more than being first. So that's kind of where it is, but that's kind of sad. So I mean, among those things were, the sequence was known first, the G protein was known first, how the system functioned, that the alpha was a GTP binding protein that's separated from beta gamma,
Yeah.
that how the G protein then activated an effector. All that was worked out first in the visual system, and beyond that, right? So rhodopsin was crystallized long before the beta receptor. So people were making, for years, people were making homology models of GPCRs based on rhodopsin. But yeah.
I remember that. I do remember that. I mean, that first rhodopsin structure in 2000 was a big deal.
Right.
And then we had to wait a long time until the next structure would come out. And as you mentioned today, you have a structure, well, might not make it into nature.
Yeah, no, unless
Might
there's
be something else.
it's
Yes.
it's got to be with, now it's like it's not necessarily with the G protein or the arrestin. It's got to be, I don't know. One thing I haven't seen, right, just if you're out there, structural people do this. There are some receptors, right? So when I was in the lab, I was working with Randy Hall, who you've spoken to. And what he did was said, well, I think receptors. He came from a neuroscience background where things are tethered, and he thought, well, receptors probably are tethered, and he identified a protein that bound through a PDZ domain to the C-tail of the beta-2 receptor. It's a protein called NERF. And so we kind of got into this kick of looking at other things that bound to PDZ domains and other receptors that have target motifs for PDZ proteins. We don't know what those do for the most. We really still don't know what those do for the most part. And there are other things. There are other proteins that are not G proteins and not arrestins and not GRKs that bind to specific receptor, not RAMPs, that bind to specific receptors. And I haven't seen a single structure for any of those yet. So hey, you guys get working on.
Yeah, all structural scientists listening.
There are other things. There are other things to look at. So just as an aside. So kind of in my own career, right? So I worked on the GRKs, a little bit on arrestins, but more on the GRKs. So rhodopsin kinase was known as an activity in the visual system long before, and it was kind of the impetus for looking for BARC, or what's called GRK2, right? And by the same token, when What's a kind of a funny story? So in the identification of GRK2 bark, right? So this is Jeff Benedict working in the Lefkowitz lab. So basically, what he showed is there was an activity that was able to phosphorylate the purified receptor in a phospholipid vesicle. Right? So you have a phospholipid vesicle, purified receptor, nothing else there. You add a lysate from a cell and hot ATP, P32 ATP, you could phosphorylate the receptor. And also, if you reconstituted that receptor with the G protein, with the GS, you could show that receptor stimulated GTPase activity of, the GP binding activity of the G protein stimulated by the receptor, right? That's what the receptor does. Could also be inhibited by this phosphorylating activity, right? So they started purifying it by analogy to the rhodopsin, thinking that there's something like rhodopsin kinase. And they showed that it's not PKA, it's not PKC, it's not any of the known, it's not affected by any of the inhibitors of the known things.
Yeah.
So they had a new activity, and they finally purified what they called bark, and now known as GRK2. But the kind of funny tidbit is during the purification step, and they note this in the paper, right? During the purification, the more pure the preparation of the kinase got, the better the specific activity was to phosphorylate the receptor, but the worse it was at desensitizing the receptor.
Interesting.
Because they were purifying away arrestin.
Oh, goodness. Yes.
So knowing about visual arrestin, Jeff got some from somebody, probably Priscilla Chesky, I don't remember who, but he got some from somebody and added it to his assay. And lo and behold, arrestin took the phosphorylated receptor and desensitized it. So then that was the impetus to then try to clone arrestin.
Yes.
So rather than try to purify it, knowing that, it must look like this other visual system component, Martin Lose in the lab used the arrestin sequence to go fishing for a somatic arrestin, which they called beta arrestin. Arrestin too, if you're,
Yes, depending on what's cool.
Yeah. So the visual system has been, very, very important touchstone. And there are papers showing that, so among the other things that Rick Sarione did in reconstituting the receptor was show that they could barely, but somewhat get transducin to work, barely for the beta-2 receptor, but for the alpha-2, which is an inhibitory receptor, right? So transducin, GT, is technically a member of the GI family, right? So for that receptor, it actually worked, right? It actually could, so that receptor could actually stimulate that G protein. So going back and forth between these systems and having those purified components available was a very important thing that gave people points of entry into some of these things, like being able to clone the arrestins. As it turned out, rhodopsin kinase was cloned after GRK2 was already identified, because it's like, it must look like GRK2.
Yeah, and then we can go back and figure out.
The other, right.
Yeah.
So it's kind of an interesting exchange between the fields. But I think that in the end, the visual signaling community's got the short end of the stick.
Yeah, they did. I just think about rhodopsin, and every time I try to do a Western blot to identify, to look at CXCR3, for example, the positive control was always purified rhodopsin. That worked so well. That was always so beautiful. Anything else I tried, forget it.
I was characterizing GRK5, and most of it was done with rhodopsin because, it was easier to get than the purified beta receptor was hard to get. Even recombinant, it was still hard to get back in those days. Mm-hmm. So yeah, I spent a lot of time in the dark room with the lights off, playing with radioactivity, getting those assays to work. But yeah, fun times.
Love it.
Okay, so a couple more kind of directions. So just one kind of funny anecdote, right? So when they clone the beta receptor, so now you can express it. So expression systems were not very developed back in 1986. So they had the gene. They didn't have the cDNA first. They had the gene, right? But so they modified it so they could express it, and they could make RNA from it using T7 RNA polymerase, or artificial RNA. And they, thinking about what expression systems were out there, they said, we used to do frogs. So this is return of the frogs. So all the first experiments that Brian Kobilka did looking at expression of the beta receptor, and then the other receptors as they were cloned and came on board, was all done by expression in xenopus oocytes, right? Yeah. Where they would make RNA from their various receptor clones and then starting to make mutations, right? Mutations were hard to make at the time.
Yeah.
And then the methods got better, but yeah, so and then starting to characterize, where on the receptor does the ligand bind, where does it couple to the G protein, why does it have the pharmacology of a beta receptor versus an alpha, and they started to make chimeras and all this kind of stuff.
Mm-hmm.
When I say they, so Brian was the one doing the assays, but his wife, Tong Sun, was apparently the master of injecting the oocytes, and she would inject hundreds of them every day in order to make enough material that oocytes are they're pretty big. I mean, they're but it takes a lot of them to do a binding assay, to do a functional assay. So basically there was a renaissance of frogs.
Yeah. I think we undervalue their usefulness as a tool, as a system.
Oh, for sure. Yeah. Mm-hmm. But then like the rest of the world, eventually everybody switched to HEK293 cells because they're terrible cells in a lot of ways. I mean, they're bad at calcium signaling. They fall off plates if you look at them.
Yes.
boy, are they easy to transfect. So that counted for a lot.
Exactly. Hence the rise of all the experiments where you only have to add stuff to it. You plate the cells, you don't look at them, and you just keep on adding things to them. And exactly.
Yeah. But anyway, so here's the Lefkowitz lab, and they're doing, literally thousands of eggs a week injecting with RNA. So they're going through T7 RNA polymerase like nobody's business. And even with Howard Hughes Medical Institute funding, Bob's like, I can't afford to do this, So they got a cDNA for the RNA polymerase and started making their own polymerase. And the joke in the lab was that somewhere in a freezer, there's $1 million worth of T7 RNA polymerase, the street value.
The street value.
Because they're doing it so much that they had to have their own source. And in fact, that when TAC polymerase came out, people would do the same thing. Labs would make their own TAC because it was expensive.
Yeah.
There was a licensing fee that you paid for doing PCR. Everybody, even academics labs, that was built into the price of TAC. So people are like, I'll just make my own.
Yeah. It's kind of a similar story in Tom Sackmar's lab at Rockefeller. So all of the GPCRs that are in the lab that I worked with as well have this 1D4 tag. And so we had the anti-1D4 antibody, and we had milligrams of it. You could really, I don't know about the street value, but it was pretty high from that perspective. And it worked really well. And then you can pull it down with it. You can detach it. It was just really an awesome tool.
Yep. So that's so just to kind of, I keep talking about various things for a long time, but I just want to go through kind of one last. It's not one experiment. It's one strand of experiments over a couple of generations of postdocs and people in other labs, to show how people characterizing receptors, how certain concepts kind of arose, right? So in the Lefkowitz lab, I told you that they had purified the alpha-1 adrenergic receptor. It was one of the three subtypes. And the person that did that was someone called Susanna Kotechian.
Yeah.
So so she started doing the same things that Brian was doing with the beta-2, just starting to make mutations. Where does the ligand bind? Why does it bind to alpha-2 ligands and not alpha-1 ligands and not beta-2 ligands, and making chimeras and all this kinds of stuff. And one of the weird things she found is she made a mutation in the third intracellular loop of the receptor, kind of near transmembrane domain 6, that converted the receptor into a receptor that was active for signaling without ligand.
Oh, I love that.
So this was the first constitutively active receptor. Right. So kind of from that beginning, having a constitutive active receptor, where did that go? What did we learn from that? So the first thing that happened was there was a medical fellow in the lab who was interested in cancer, a guy named Lee Allen, and he expressed this constitutively active receptor, which activated phospholipase C beta, so basically turned on calcium signaling through diacylglycerol. And did this 3T3 cell cancer assay where you look for cells that overgrow and make colonies. And it turns out this receptor is, they called it an oncogene because it was positive in this assay. Basically, it caused the cells to grow without stopping. So that's kind of the first GPCR as a potential tumor thing. And that's
Interesting.
Yeah.
Please, I just want to add, when I think about GPCRs in cancer, it took a long time and it's still not that well. Okay, so I think there was the GPCR field and people working on GPCRs in general, and then there is the cancer field, and the connection between the two is not as strong as it could be considering the role of GPCRs in cancer.
Yes, which is modulatory. I mean, they, rightly so. At the beginning, cancer was focused on individual mutation drivers.
Yes.
But I think we're at the point now where I think it's very clear that one, it takes multiple hits, but two, equally important to having a driver mutation. So a cancer cell is in communication with its microenvironment. So it has stromal cells around, normal cells around it, stromal cells, that it is converting to a cancer cell phenotype, whether it's through signals going back and forth through things like GPCRs or potentially through things like exosomes that are being secreted from the cell. But it reprograms the cells around it to create the environment it wants. And if you can interrupt that, that can be a therapeutic, even despite the fact that the cancer cell has a driver, because you can deprive that cancer cell of the support and community that it wants around it, right? So, and I think so. There's certainly a role for GPCRs in those kind of processes, and we'll see kind of where that goes. So they had this constitutively active receptor. So at a single point mutation, they changed one amino acid. And there was a student in the lab, Mike Chelsberg. I think was he a postdoc or a student? I don't remember now. But anyways, and what he did is he mutated that single residue in that one receptor to every single amino acid, and then characterized their activity. And it turned out that the natural amino acid was the lowest activity of all. Anything else you changed it to made the activity go up. Some things more than others, but every other, So basically, this receptor had been selected evolutionarily for that particular critical residue. It had to be this to have a low basal activity, and anything else you changed it to was going to have a higher basal activity. Which
so
is really critical.
No wonder that residue was there. It was there for a reason. Wow.
Yeah. And I think I'm not that much of a structural biologist, but I think if you go back and you look at what is that doing in the structure relative to G protein activation, it's going to be one of those very critical residues. Yeah. Okay. So now you've got this receptor that's tuned to be with low activity, and people still think, well, the receptor is off until you turn it on, like a light switch. That's still kind of the predominant thinking, but that's about to change. So then a student in the lab, Philippe Samama, made the equivalent mutation in the beta-2 adrenergic receptor, making the first constitutively active beta-2 receptor. It's coupled to GS. Of course, it's not going to be an oncogene because it's cyclic AMP cytostatic, so it's fine. But it's constitutively active, and he started characterizing it, and got some kind of unexpected results in looking at it. So two aspects of unexpected results. One, the way it acted was not quite what you would expect based on what was called the ternary complex model, right? Which is something that Andrew de Leon had developed when he was in Bob's lab years before. So Philippe started working with Tommaso Costa, and Philippe doing the experiments and Costa doing the modeling. And so they have a paper on the extended ternary complex model. And I think if you look at Google Scholar and you look for Lefkowitz, he's got all kinds of reviews that I got highly cited reviews. The first actual paper, the highest citation is this, because it really changed the way people think about how receptor allostery works, right? So if you change the ligand, you can change the way the receptor works. If you change the G protein, you can change the way the receptor works. But if you change the receptor, either by changing its level or changing its structure through other allosteric means, whether it be an allosteric regulator or a mutation,
Yeah.
you change the way the receptor works. And that's really the first theoretical explanation for that.
Wow. Love
Right.
it.
So they published that in 93. And now the interesting thing was, so now people started to really purify receptors from other sources. And among them was people were expressing receptors in insect cells, baculovirus and SF9 cells.
Mm-hmm.
And one of the people doing that was Michel Bouvier, right? So they were expressing all these receptors in SF9 cells, making membranes that had tons and tons of receptor. They started a company selling them to pharmaceutical companies for doing assays with. And one of the things they noticed is that if you have a receptor and you overexpress it high enough, it changes the basal activity. So you don't have to be mutated to be constitutively active, right? From a theoretical framework, if you have more receptor in the system, you push more, there's more of it in the active state, you see more activity, right?
Yeah.
But this really showed that's true. The more you increase the level of the receptor, the more activity you get out of the system without a ligand around, right?
Yeah.
And having identified that, now what Michel did was like, well, let's see if we can antagonize this. So they dumped various antagonists on it. And that was where, the idea of inverse agonism was kind of out there beforehand, but this really, definitively showed it that if there are ligands that do nothing to the active state, so you add them to this constitutively active receptor and it does nothing. And there are other ones that are actually inverse agonists or negative agonists that decrease the basal activity because they shift the equilibrium of the receptor from active state to inactive state. Yeah. So
I think it's wonderful, and I wonder, I may be mistaken, but I think this was work well done in Michel's lab, but by Graciela Pinheiro, if I remember correctly, from one of our podcasts, actually third or fourth podcast episode we had.
Yeah. So that was in like, what 1984, 1985. No, 94. Yeah.
Yeah.
94. So the other kind of funny thing that happened is, so in the Lefkowitz lab, we got medical fellows. So a medical fellow with not a lot of research experiment came into the lab. Very nice guy, Carmelo Milano, and he was a cardiology surgeon. So he's not like he's chief cardiac surgeon at Duke, but he wanted to learn a little bit of research. So he was given the job of making a transgenic mouse expressing the constitutively active beta-2 receptor mutant in the heart. Okay, so someone hands him, we got the promoter from myosin heavy chain cardiac version or something, and then he did all the manipulations to put the receptor driven by that promoter, made the transgenic mouse. Never checked that the clone that someone handed him was in fact the mutated receptor. So it turned out, so then now they got the mouse, and it's got this amazing phenotype. The heart beats like, the heartbeat on a mouse is like 200 or 300 beats per minute. This is like 500-600. It was crazy. It was like on overdrive all the time, this mouse. But when they started looking at it, there are things about it that the pharmacology didn't fit right with the constitutively active receptor. And they realized, oh my gosh, this is the wild type.
Oh, wow.
So someone had handed him a tube and said, here's the mutant. And just like the wild type, it's got the same restriction sites. You cut it out by hand, it's the right size, everything looks good. And this is in the days when sequencing was, expensive, slow, radioactive. You read the bands on the ladder on a gel. So it wasn't sequenced. So eventually he made the constitutively active version of the mouse too. P.S. The phenotype in the wild type was 1,000 times better than the constitutively active one, because the wild type mouse, because it's expressed highly, is constitutively active on its own, right? Of course. And the constitutively active mutant, because it's constitutively active, it spends more time in the active state, it's less stable, it gets degraded. So in the end, the level of expression in the transgenic mouse with the constitutively active mutant was just a little bit above normal. And there was almost no phenotype, very subtle phenotype. So the phenotype in the wild type, it had like 500 times the normal expression, crazy levels. And that characterizing that mouse got them a paper in Science. So this mistake
Yeah.
That's about paper and science. And of course, all the postdocs in the lab were aghast, and it's like, oh my God, I'm going to sequence everything that anyone ever hands me. And this is very good advice. No matter you buy it from AdGene, you get it from somebody in the lab. Sequence, sequence, sequence. You make sure that.
Always,
Oh my gosh.
always. We
Yeah.
had that happen to one of my a colleague of mine during my master's. He spent the entire summer trying to detect a protein at the cell surface. And it was a construct that we received from a collaborator. And 3 months in, we realized that actually it didn't have it didn't have the transmembrane domain. It was a single transmembrane. So
Mhm.
the protein was expressed, but he every time he would change the media to do the cell surface staining, he was washing it off.
Yep.
Uh, this person is not in research anymore. I think
Well,
because that was a frustrating.
we've all been there and done things like that, but it's
So,
usually
but
we don't make
yes,
a mouse doing
yes.
it anyway. But it turned out well for him. He got a science paper. But the other thing is, right, so now they had this mouse that had the wild type receptor, has this great phenotype. They had the mouse that had the constitutive active receptor that was unstable. And what they did was, now they said, well, there was this hint in the literature. There's this other literature on like the nephrogenic diabetes insipidus with the V2 vasopressin receptor.
Yes.
It was actually Lutz Birnbaumer's wife, Marielle Birnbaumer, was a big person in that area. But basically, what they found is that if you add a ligand, you can stabilize a misfolded receptor and get it to go to the cell surface, right? So in the case of this constitutively active mutant beta receptor, if you add an antagonist to the receptor, it now gets to the cell surface and the levels are higher. And actually, the activity of the receptor goes up with the antagonist because there's just so much more receptor there that's active, despite the fact that there's a ligand that's supposed to be an antagonist on it, right? So all of these things came from one mutation.
Yeah. Yeah. I think I've always loved this field, but listening to these stories just kind of puts it into perspective as to how cool are these proteins and how one mutation, one mistake leads to discovering these phenotypes and better understanding these receptors.
Great. Well, that's kind of, I could keep on going, but I think we've been talking a while, and that's kind of
That's
a good dose of GPCR history.
I think so too. I think it's a good dose. But as I mentioned, I know we're still recording because as mentioned before we hit record, you're always welcome. Anytime you want to share more history, I'm pretty sure that all our audience will enjoy hearing more about these because these are the things that either we don't think about on a daily basis, or we don't even know that these things existed.
Well, if you happen to be in one of the labs where some of these things happened, you'll hear some of the stories.
Exactly.
But if you're not, again, if you wanted to go back and reconstruct the history of the field by searching PubMed, you're going to have a hard time because of name changes and
Yeah.
yeah. So it gets kind of. I was like, you want CXCR7. You happen to, I mentioned RDC orphan,
Yes.
but you're like, you knew, oh, RDC, I think it's RDC1 is CXCR7.
Seven. Yes. Which is
But
now
there are
not
all
even
these
called.
alternative names that it just gets, at some point it gets hard to search back.
Exactly.
So,
Exactly. And I think now it's called ACKR3. So
well,
atypical
fine.
chemokine receptor 3, which is another way of calling it. And as things evolve, and you mentioned, being second and having the beta adrenergic receptor cloned, as things evolve and more receptors are discovered, the nomenclature changes. Yes, we capture it in an IUFR publication, but then you can't go back into the initial publication and say, well,
You change
actually,
that. Yeah.
this is not how we called it, or this is now how we're calling it right now.
Right.
Thank you, Richard. This was an
Oh, that's fun.
amazing,
Yeah, thank you. Hopefully people
fun.
enjoy it.
Yeah, I did. I did. I'm pretty sure people are going to enjoy it as well. And thank you for taking the time to actually go back in time and read these papers and look up this information. Oh,
Well, I told you kind of more privately at the beginning,
yeah.
I at some point several years ago, I'd started to write a book on GPCR. So I was like the history of the GPCRs and the kind of state of the field, because there is no such a thing anymore, right? So my most highly cited review is one that I wrote with another postdoc in Bob's lab, Kristen Pierce, and Bob, and it is the last review that actually covers G protein coupled receptors, and it gets cited every week. I mean, but the field is too big to
Yeah.
fit in one thing, so it kind of needs a book, and so somebody needs to write the book. But I don't
Yes.
have the
Yes.
time. But I had started
Yeah.
at one point, and so I kind of started to refresh my memory for all these stories that I'd heard over the years.
Yeah. I was just thinking about that. Somebody or people need to put their heads together and write the book,
Well, I mean,
because I think it would be really cool. I don't know who has the time and who can help do
right,
that, but I think,
or who reads books
well,
anymore.
I don't know, audiobooks. We can have,
Not true.
we can have
A famous
either
actor.
people who made the discoveries or people who worked in the labs where these discoveries made read chapters. I think it could be a really, obviously it's a lot of work, but if we set that aside, it would be really nice to have a book
Mm,
that captures the history of the field and also have it in an audio version,
right.
where we have select celebrities of our field read chapters. Could be really cool.
Which is why it's sad you never got to talk to Mark.
Yes.
But moving
Yeah.
forward, anytime you get somebody with a lot of gray hair, you can add to your questions. Hey, what cool stories do you know about what happened
Yeah.
back in the day? And who knows what you're going to turn out.
Absolutely. That's a wonderful idea. And I was thinking about, it's been 3 years, and I think we need to upgrade our questions a little bit. They're pretty wide, but you make a great point. Thank you for the feedback. Thank you. I really appreciate it. Thank you for your time. I really enjoyed talking to you.
Oh, yeah. Well, thank you for doing this. It's been, I mean, I enjoy listening to him too. So it's great.
Amazing. All right, we're gonna stop recording. Let people wonder what we're talking about right after. I'll talk to you soon. Thank you, Richard.
Thank you.
Can I ask you for a favor? Please subscribe to our Dr. GPCR YouTube channel. Many of you come back regularly to watch our videos, like the monthly video edition of the newsletter, but aren't subscribed. Having more subscribers will help us get you more content. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank you for listening, thank our guest for joining us, and also would like to take a moment to thank our team members, Attila, Ines, Montserrat, Ivana, Andreina, and Balint. A huge thank you to our Dr. GPCR Ecosystem partners, Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. If you like our podcast, leave us a 5-star review on Apple Podcasts. You can also leave us a testimonial at drgpcr.com/testimonials. You can also subscribe to our monthly newsletter delivered directly to your inbox by visiting ecosystem.drgpcr.com/newsletter-sign-up. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.